We're starting to record now. Again, my name is Nelly Iredale. I'm the sounding board chair. This is Oceanids' 68th year, which is an official support group of the university. The sounding board is a joint effort of the faculty club and Oceanids. Our honorary chair is Tuszynski Phd, the associate of the chancellor, and our president, Nigella Hilgert. In Oceanids, we raise money for grants and awards for students, support other UCSD events and activities such as the student-run free clinic. We offer many events for our members, and Sounding Board is one of these events. We would love to have you as a member, and I encourage you to join. It's only $35 per year. We are happy to see you now via Zoom. We have this event every first Thursday of the month throughout the academic year, except for January. And we have this special presentation today in the month of June. Now, I would like to introduce to you our speaker, Dr. Mark Kucinski, Ph.D. He's the director of the UCSD Translational Neurosciences Institute, Distinguished Professor of Neurosciences. A little about his bio. Dr. Mark Kucinski is a physician scientist exploring the topics of spinal cord injury, degenerative disorders of the nervous system, and fundamental mechanisms underlying motor learning and memory. Dr. Tuszynski obtained his Bachelor of Science and MD degrees at the University of Minnesota and completed residency training in neurology at Cornell University Medical Center, the New York Hospital. He then earned a PhD in neuroscience at UCSD and has been a faculty member since 1991. He is currently the director of the Center of Neural Repair and founding director of the UCSD Translational Neuroscience Institute. Dr. Tuszynski has over 300 publications, including 3 books. The overarching goal of his research effort is to develop effective therapies for untreatable neurological disorders. Dr. Tuszynski performed the first human clinical trial of gene delivery in the adult central nervous system, nerve growth factor gene therapy for Alzheimer's disease. Additional clinical trials of growth factor gene therapy followed in Parkinson's disease and a current clinical trial in Alzheimer's disease. He has received 15 awards for his research. His research is supported by the NIH, the Veterans Administration, and several foundations. He will talk to us about gene therapy for Alzheimer's disease. We will keep being muted, and Professor Tuszynski is able to take questions as he goes along. So you could raise your hand digitally, or we will be checking the audience, and just raise your hand if you don't know how to do it digitally, and we will try to catch you. Professor Tuszynski, welcome. Thank you so much for being here. Please go ahead.
Hi, thank you for having me. It's nice to see everybody. I understand many of you are UCSD faculty and former faculty, so it's really a pleasure to be able to talk to you today. And I'd like to encourage anyone to do ask questions if you like while we go along. Questions often come up, and I'm happy to address them. So I will start screen sharing, and I am indeed going to tell you about one of our 3 research programs. Oops, but not starting in the middle, hopefully, but starting at the beginning. This will be our story on gene therapy for neurological disorders. And in particular, a current clinical program we have that's called a phase 1 clinical trial of AAV2 BDNF gene therapy in Alzheimer's disease and mild cognitive impairment. Before diving into that, I'd like to give a little background about gene therapy and why this matters. So let's start there. So gene therapy refers to delivering therapeutic genes to the nervous system. And there are two general approaches to this. One can either replace a deficient gene that's a product of a genetic disorder like Tay-Sachs disease, or one can, on the background of a genetically normal nervous system in somebody who has an acquired disease like Alzheimer's disease, one can use gene therapy just as a tool. It's just a tool for delivering something potentially therapeutic into the brain. And there are currently two efforts in Alzheimer's disease at gene therapy. One is ours, in which we're delivering growth factors of the brain, and I'll tell you about that shortly. And there's another gene therapy trial currently underway in Alzheimer's disease also, in which the investigators there at Cornell are attempting to provide patients who have a form of this ApoE4 gene that is associated with earlier onset of Alzheimer's disease. They're trying to give these patients a replacement more normal gene called ApoE2. And I won't say more about that. I know that you probably don't know what we're talking about here, but if you're interested in Alzheimer's disease and gene therapy, one can look up this trial, again, being run at Cornell. And so gene therapy is something that the world of science has recognized as a great potential benefit to the treatment of human disease for about 30 years, but really hasn't shown any success until just the last couple of years. And there are currently 2 approved gene therapies for human disease in the United States. There's another one in Europe for a lipid disease, but that one wasn't approved here based on the data that was presented. So the first gene therapy to be approved in this country was replacing a deficient gene as a result of a genetic disorder. And this was in a form of retinitis pigmentosa called Leber's congenital amaurosis. So as you may know, unfortunately, patients with retinitis pigmentosa start to lose their vision, often in their teens, and can be entirely blind by their 20s. And some of these forms, and there are many genetic mutations that are associated with retinitis pigmentosa. And one of them is this Leber's congenital amaurosis, in which a specific gene called the RPE65 gene has a mutation that leads to this progressive degeneration. So in this first approved gene therapy, what the investigators did was take a gene therapy virus vector, as it's called, and I'll explain that more in a second, but they used it to shuttle into the eye a normal replacement gene for this mutated gene. So in this case, a needle is placed through the white part of the eye, the sclera, and introduced into the back of the eye where the degenerating cells are that the so-called photoreceptors that perceive light. And a little bleb is created. In fact, a retinal detachment is created in which the virus is delivering a normal copy of the gene to these cells. And this bleb, this retinal detachment, resolves on its own after a few days. And now these cells have the chance to take in this gene. And as it so happens, this ends up improving vision in these patients. This was a new clinical trial, and there were really no good outcome measures for measuring some partial restoration of vision. So these investigators developed a spatial navigation system to see if the patients could actually navigate this complex environment better, and they could. And they subjectively reported some improvement in vision. So that's kind of revolutionary. I mean, you know, the era of molecular biology has been on us. And by the era of molecular biology, I mean our knowledge about genes and messenger RNA and how proteins function in the cell. You know, this just came into neuroscience when I began as a neuroscientist 30 years ago at UCSD in the lab of Rusty Gage, who's now the president of the Salk Institute. But molecular biology was just developing then. And this is really the culmination of going from research to human benefit in terms of gene therapy. So this was revolutionary in 2017. And then another treatment was approved in 2019. And this is not an ocular disease, but it's a pure neurological disease. So there's a syndrome called spinal muscular atrophy in which human infants are born weak. And over the next 18 to 24 months, most of these children go on to die because the diaphragm also becomes weak and they can no longer breathe. and they can be kept alive beyond that by being on respirators, but it's a miserable disease. And children uniformly died of this, and now they live. Now they can receive a replacement gene for this mutated gene in spinal muscular atrophy called the SMN1 gene. So 1 in 6,000 children are born with this spinal muscular atrophy. It's like ALS or Lou Gehrig's disease in infants. 1 in 40 people are carriers for this genetic mutation, so it's not that rare. And in a landmark achievement, the investigators in the study delivered a normal copy of this SMN1 gene to children with spinal muscular atrophy. And the results were really quite astonishing. These are the survival curves of the individual patients in the trial. So the red line indicates, sorry, it's not a red line, it's a green line. That indicates basically here that by 20 months, all of these children were still alive. And continuing to monitor these children over time, they're still alive, and many of them are gaining motor milestones. And this was when, in the absence of this gene therapy, all of these children would die of respiratory failure. So this is just an astonishing breakthrough and is vitally important. So might we be able to take some of this success and move it to other diseases? And that's part of the effort we'll be talking about today. Let's talk a little bit, though, for a moment about how one does gene therapy. So in most cases of attempting to treat disease with gene therapy, we use a tool called the adeno-associated virus. So this is a virus that is a respiratory virus normally in humans. It has this viral coat. We're used to looking at pictures like this now with COVID. This is the viral capsid, as it's called, which is made up of proteins that allow the cell to attach the cell and enter the cell normally. We take this normal virus, this so-called wild type virus, and we gut it of all of its natural genes. So it's just a shell. And what we do is replace those viral genes with our therapeutic gene. And it cannot cause disease because it doesn't express the wild type genes. And even this wild type gene that does contain the viral genes on its own doesn't cause disease. It normally needs a different virus, an adenovirus, to cause disease in humans. So there are sort of two levels of safety built in to these gene therapy methods. One is that we gut the disease-causing genes and remove them. And secondly, we don't introduce the gene therapy with this adenovirus. So it can't replicate on its own. It's impotent. There are several natural types of this adeno-associated virus. They're numbered from 1 through 11, and numerous additional designer forms of this that have been developed for gene therapy. But the most common and the most frequently studied in clinical trials is a type called AAV2. I've just actually talked about this, so I'll skip it. So when we've taken, this is a diagram of the normal set of genes in this AAV2 gene therapy vector. And normally there would be a bunch of the viral genes right in here in between what are called the 5' and 3' intermediate terminal repeats. And what we do is we take them all out and gives us space of about 4,500 base pairs in which we can insert our therapeutic gene. And so this is a vector of what our therapeutic gene looks like. We have these ITRs that are the initiation sites for trying to express a gene. We put in something called a promoter that turns on the gene. And then we insert our therapeutic gene that we call the transgene. And the identity of the gene we're trying to express depends on the nature of the disease. In spinal muscular atrophy, it's a normal copy of the mutated SMN1 gene. In our effort in Alzheimer's disease, this is a gene for a growth factor of the nervous system that I'll tell you about more shortly. So it's actually rather simple. And so there are several current gene therapy trials that are ongoing at various institutions around the country and around the world, targeting different diseases. So there are several efforts in Parkinson's disease. One of these is a completed study. It delivered a growth factor gene called nurturin that is meant to prevent the death of the cells that are vulnerable in Parkinson's disease. That trial was done by a company I founded 20 years ago. I no longer have a company. I have no financial conflicts of interest now. This trial ultimately failed for reasons that we'll get into later. But the effort in Parkinson's disease with growth factors is continuing. There's a current trial at Ohio State delivering another growth factor gene for cells that are vulnerable in Parkinson's disease. Then there's another clinical trial that is not a growth factor, that is delivering an enzyme to increase the synthesis of the neurotransmitter in the brain that becomes deficient during Parkinson's disease. And the name of that enzyme is AADC, short for aromatic acid decarboxylase. Then separate from Parkinson's disease, there's another disease like Parkinson's, but again, it starts in infancy. This is called congenital AADC deficiency. And again, they're trying to replace this mutated gene in 2 clinical trials being run out of Ohio State. Then we have our efforts in Alzheimer's disease, as mentioned, our growth factor trial, and then the trial at Cornell. And then in spinal muscular atrophy, even though the trial I just told you about was a smashing success, the thought is that they could even improve it further by using a different route of delivery of the gene therapy. So the first trial was successful based on peripheral IV administration. This is seeing whether higher doses of the replacement gene can be achieved by doing a spinal tap and giving the gene therapy vector that way. Still pretty simple though. And then there are a number of metabolic diseases that typically affect people in infancy. These are the various mucopolysaccharidoses. They're listed here. One is called Batten's disease, another is called metachromatic leukodystrophy. But there are all efforts underway in these congenital diseases to provide a normal copy of the mutated gene. Again, the first breakthrough came in spinal muscular atrophy, and the importance of that, the milestone that represents, cannot be exaggerated. A once uniformly fatal disorder just 2 years ago, it's now treatable, and these efforts are ongoing in a number of other diseases. But there are some issues, just speaking of the gene therapy world in general. One is that with the technology we have today, we turn on the therapeutic gene, but we actually can't turn it off. So what if there was a problem? And there's unlikely to be a problem with the expressed gene if you're replacing a deficient mutated gene. But what if you're delivering a potentially therapeutic gene like a growth factor or ApoE2? What if providing an extra amount of it could actually be a problem? If it is a problem, you can't turn it off once you've turned it on. There's no way of really stopping this. And so there's an effort to try and develop means to regulate and turn off gene expression in the future. These are being developed, but none of them are effective now and ready for clinical application. Another issue is identifying the optimal route of administration. Do you give the gene therapy systemically where it infects every cell of the body potentially? For treating a disorder of the brain, can you give it just as a simple spinal tap? Or do you actually have to provide it directly into the brain, what's called intraparenchymally? And just what the optimal route is depends on the nature of the disease and the payload gene that you're delivering. And this is particularly an issue with the growth factors that we study, as I'll tell you in a few minutes. So the IV and intrathecal route are often used when there's widespread degeneration all over the nervous system. In the case of Alzheimer's disease, that's not the case. The degeneration is mostly up here in the superficial, in the topmost part of the brain, the cerebral cortex. So if you're delivering a gene that might have off-target effects, well, maybe that's not good if you're expressing it everywhere. And then there's ongoing research about just what the best viruses to accomplish gene therapy. Currently, the 2 that are used clinically are these AAVs that I've talked about, and another type is a HIV-related virus called a lentivirus, in which, once again, many of the disease-causing genes are removed. But there are different types of these AAV vectors. There are what are called artificial and hybrid vectors, and that's all the topic of much general research in the field of gene therapy. And then finally, there's the issue of cost, and this really speaks to an ethical issue. Luxturna is the treatment for retinitis pigmentosa, and the drug company that sells this is selling it for the astonishing price of $425,000 per eye. And of course, we all have two eyes, so the real cost is $950,000 per treatment. And it isn't clear whether people might need a subsequent treatment, whether the amount of gene expressed with a single treatment will be enough. Zolgensma is the name of the gene therapy for spinal muscular atrophy in infants, and its astonishing cost is $2.1 million. And when the number of patients is small, then drug companies are potentially willing to pay prices like this. But if there's a disease that affects thousands or tens of thousands or millions of people like Alzheimer's disease, these costs become an important issue. And because I did found a gene therapy company in the past, again, I don't have one now, I have no conflict of interest, but I had one in the past. And so I know what it costs to make these vectors. To make the vectors in the amounts we need them to treat the brain, the cost of the vector, the actual product, is about $1,000. So there is a huge profit margin built into this. And it's just not necessary to charge this much for more common disorders like Alzheimer's disease. So were one to identify a useful therapy for Alzheimer's disease, likely the cost would be much less. So let me pause there for a moment and just see if anyone has any questions about that before I go now into more detail on what we're trying to do in Alzheimer's disease. And Nellie, I'll sort of let you guide things, if anything pops up, just let me know. And if not, I'll just keep going.
Thank you very much. I don't have anything on the chat and I don't have raised hands, so we can keep going. Thank you.
So I'd like to tell you now about what we're trying to do in Alzheimer's disease. And again, this is the name of our program. We're delivering this AAV2 type vector to deliver a growth factor called BDNF into the brain in patients with early Alzheimer's disease and mild cognitive impairment. So Alzheimer's disease is the most common age-related neurodegenerative disorder that at the end of the disease results in severe cognitive decline. And there are over 20 million people affected with this worldwide, and that number will double in 25 years because this is an age-related disease. And the longer people live, the more people there are more people that are vulnerable to developing Alzheimer's disease. And sadly, it remains the case that there are no broadly effective therapies for Alzheimer's disease. There are no therapies that slow cognitive decline. There are 2 approved drugs. They have relatively mild effects that are temporary in the early disease. So there's a great need to develop therapies that can slow the course of Alzheimer's disease or even potentially improve cognition. Alzheimer's disease is caused by a combination of the formation of a buildup of a protein in the brain called amyloid plaques. There's also degeneration in the structure of neurons, the cognitive cells of the brain. Their internal structure undergoes degeneration in what are called neurofibrillary tangles. And as a separate risk factor that I should really add to this list is, again, aging. There are just age-related changes in the brain, some atrophy, that if you don't have problems with plaques and tangles, still allow you to have normal cognitive function. But when combined, when the age-related changes in the nervous system are combined with plaques and tangles, then one starts to suffer cognitive decline. And this cognitive decline first begins with the loss of the connections between cells in the brain, the synapses, and ultimately results in the death of cells in the brain. So let me spend a little more time on this. So the candidate therapies in Alzheimer's disease are trying to slow degeneration. And because amyloid is one of the chief components of pathology, some therapies are trying to degrade amyloid or remove amyloid from the nervous system. And there have been a number of these trials. Billions of dollars have been spent on these clinical trials in tens of thousands of patients. And none of them have shown clear efficacy. It's possible that there was a signal of a benefit in patients with very early Alzheimer's disease. And one drug company is pushing the FDA to approve its drug based on this relatively weak clinical data. Experts in the Alzheimer's disease field are opposed to the FDA approving this because there's not enough evidence that it does anything. And the experts in the field are suggesting to the FDA that another clinical trial should be done in patients with early Alzheimer's disease to determine convincingly whether this is an effective therapy. So it's somewhat controversial right now. The other pathological hallmark were those neurofibrillary tangles, and they're associated with a type of protein called tau. And so there are anti-tau therapies that are now undergoing clinical testing in Alzheimer's disease. It's really early in the course of testing this candidate mechanism, so we really don't have a sense yet about whether it will be effective. There have been a couple of early results from trials, and they haven't been clearly positive. But who knows? And testing in larger number of patients, something may emerge. But this lack of therapies targeting the key pathologic components just really highlights the importance, the need for new alternative treatment strategies. So our strategy is to take this growth factor of the nervous system called BDNF, and we are trying to test the hypothesis, test the idea that this BDNF will protect and enhance the function of cells in the brain that serve memory. Those are called entorhinal cortex cells. So we target this growth factor toward this memory circuitry of the brain and are exploring whether it will protect and enhance the function of these cells, representing a potential neuroprotective strategy in early Alzheimer's disease and its prodromal state, mild cognitive impairment. So for up to 5 years before people develop clear Alzheimer's disease, they suffer some memory loss. They suffer a little bit of cognitive impairment that differs from just normal absent-mindedness that occurs as we all grow older. And when one can demonstrate that on a neurological examination, then we diagnose that as mild cognitive impairment. And as I say, that's sort of a pre-runner to developing the actual diagnosis of Alzheimer's disease. So our movement of BDNF forward as a candidate therapy is based on animal studies in so-called amyloid mice, in aged rats, in injured rats, in aged monkeys, and in injured monkeys that all consistently showed that this growth factor, BDNF, prevents the death of neurons, of the key thinking cell of the brain, that this therapy builds new connections, new synapses between cells in the brain. It also improves what we call the transcriptional state of the cell. If you look at all the genes that are perturbed by amyloid disease or by Alzheimer's disease, a lot of them are going down with the disease, and this BDNF restores much of this gene-related expression. And then again, in mice, rats, and monkeys, the delivery of BDNF improves cognitive outcomes. So this represents a potential new class of therapy that targets a novel disease mechanism than the amyloid and tau. This is an entirely different way of trying to approach the disease. So this program with BDNF, item number 2 here, represents actually our 5th effort of gene therapy nervous system. We did 2 clinical trials with nerve growth factor in Alzheimer's disease. We did 2 clinical trials with this other gene therapy in Alzheimer's or in Parkinson's disease, and they both failed, and I'll show you why. And our current effort attempts to take advantage of newer technology and a new growth factor that we think will be a more potent growth factor in this clinical effort. So what are nervous system growth factors? So these are natural proteins of the brain. They are naturally made by the brain during the development of the nervous system during embryonic and fetal time periods. These growth factors support the survival of cells in the brain, and they continue to have an important impact on the brain after development, throughout adulthood and into aging. These growth factors normally support the function of the intact adult nervous system. They improve the excitability of cells in the brain. They help to maintain the functional state of the cell. And then if we take them to animal models of degeneration of the brain, they potently prevent the death of cells in the brain, and they enhance the function of cells in models of brain disease. And I'll show you that now, if I can get my computer to advance. This shows the signaling of growth factors in the brain. This is a complex slide that's really biochemistry. I won't go through this in much detail other than to say growth factors are present out here in the outside. This is the inside of the cell, and this is the outside of the cell. Growth factors are made by other cells in the brain. They're secreted into the space, into the fluid between cells in the brain. And they bind to receptors on the cell surface. And when they bind, they activate signaling in the cell through one pathway here that's called the MEK-MAP kinase Krebs pathway to activate the functional state of the cell. They also activate a second pathway called the PI3 kinase and AKT pathways that reduce the death of cells. So they act both at the molecular biochemical level to activate the functional state of cells and to reduce the amount of cell death. And this is how we got to learn of their function. So this is a diagram that shows that there's a population of cells here in the part of the brain called MS. This stands for medial septal nucleus. And this is a diagram of the rat brain. So in the rat brain, there are these cells here deep in the brain. They send their connections called axons into the memory circuitry of the brain called the hippocampus. And if you cut these axons as they project to the hippocampus here, if you do a lesion in the brain, then these cells back here in the medial septal nucleus, they shrink down, they undergo atrophy, and then they die. These cells use a neurotransmitter, a neurochemical called acetylcholine. And in projecting here to the memory circuitry of the brain, they play an important role in just modulating the excitability and the functional state of neurons involved in memory. So when you do a lesion like this, there's an impairment. And this is what that cell loss looks like. So this again is the rat brain. This is a 40 micron thick section. So it's a section about the thickness of a hair that shows where these cells are in this deep part of the brain. Each one of these little black dots is a cell shown here bigger at higher magnification that is a cell type that happens to degenerate in Alzheimer's disease. But these are rats, they don't have Alzheimer's disease, and this is a normal rat. And I'm showing in the box on the left where these cells normally sit in the brain. If we do that injury I just showed you, if we do this cut here, then on just the left side of the brain where we do the cut, these cells degenerate and die. But now look here, if you give the nerve growth factor protein, then these cells look like they're surviving. And if you quantify this effect, this is what you see. Without the growth factor, you lose about 60% of the cells with no treatment compared to the normal number. But now if you give the growth factor, about 90% of the cells survive. You know, in science, you often have effects that are statistically significant, but not that great in magnitude. Well, this is a big magnitude effect when you're rescuing 90% of the cells. And this is consistently found whether you give these rats nerve growth factor from mice, give them the human version of nerve growth factor, given in low or high concentrations, it's potent. It prevents the death of these cells. Well, these cells that we're talking about undergo degeneration in Alzheimer's disease. They're one of the components of cell loss. So it occurred to everybody in the field 20 years ago when this was discovered that, oh, maybe this could be a useful therapy in Alzheimer's disease. So the first thing we did was to say, well, let's get closer to humans than mice and rats. Let's see whether this is actually effective in a primate brain. So in monkeys, we performed the same kind of lesion. We went in with a scalpel, we just cut that connection. Monkeys were unaffected. You know, they still functioned as far as one could tell normally. If you did detailed memory testing, they would have a bit of an impairment, but functionally they seemed okay. So, you know, these are not harmful lesions to the monkey other than causing some memory loss. And here we cut the connection on the right side of the brain. So here are the normal cells on the left side of the brain. Each black dot, again, is a cell. And again, you have a sense that there's an attenuation on the injured side of the brain. But now if we give the nerve growth factor, then look at this. You have a sense that there's some protection. And if you quantify this effect, in monkeys now, with this kind of an injury, about 75% of the cells die without the growth factor, and about 80% survive with the growth factor. So again, these are potent, striking effects. So a lot more research was done. Other investigator groups showed similar findings. Some of them are listed below. And this is just one more example of the effect we see. Remember I said these cells project up here to the brain, and this is where their axons are, where they make contact with the cell circuits in the memory part of the brain, the hippocampus, and play a role in memory. And this is what these lines are, those axons, those connections reaching the cortex and the hippocampus. And this is they're what they look like in a normal young monkey. With aging in the monkey, there's a reduction of these inputs to the cortex. And now if we give the aged monkey the nerve growth factor through gene therapy, again, you look at this and you have a sense, oh yeah, it looks like there's some improvement. And if you quantify this and look at the levels of these inputs in young monkeys, there's a loss in aged monkeys, and the nerve growth factor restored that entirely. Something analogous occurs in human Alzheimer's disease. This is a normal aged human, and this is the Alzheimer's brain. This, again, these are these connections, the so-called cholinergic axons up here in the cortex in Alzheimer's disease. And so in moving this program to humans, we said, well, if we can restore it to something like this, then might we improve memory? And after doing extensive testing in rats and monkeys and showing that this was safe, the FDA gave us approval to move forward with this into humans. Now, again, this is not BDNF. I'm starting with the earlier history where we did the first clinical trial using this other growth factor called nerve growth factor on this cell population in the brain. And so how are we going to deliver this nerve growth factor? Well, the nerve growth factor, it's a protein. So it's a big molecule and it's polar. It has a charge on it. And when things are big and are charged, they tend not to get into the brain very well because there's a barrier in the brain called the blood-brain barrier that excludes many large and polar proteins like this one. So to reach the brain, it has to be administered directly into the brain to exert these potent effects on their neuronal targets. But it turns out that with this growth factor, nerve growth factor, if you spread it all over the brain, it does cause side effects. It causes weight loss, it causes pain, and it causes some cells to migrate into the brain and surround the base of the brain in this layer of expanding cells. None of those things are good. If you have a disease, you don't want to be losing weight. If you certainly don't want to suffer from pain as a result of your treatment. And why did the pain occur? It occurred because other cell types in the nervous system respond to the nerve growth factor. The fact that pain was developing was a sign of the response of the body to the nerve growth factor. So for us to try and make this a therapy to bring into humans, we can't spread it all over the nervous system. We have to get it just to the area where the cells are degenerating. And that's kind of a challenging thing to do. One way to do that is gene therapy into the brain. So if we put a needle into the brain and restricted the delivery to the area of the brain where the cells are degenerating, then it doesn't spread all over the nervous system and it doesn't cause the side effects. And we showed that it doesn't in rats and monkeys before moving it to humans. The other way you could potentially locally deliver it into the brain would be to do a pump and pump in the nerve growth factor protein itself. And we considered both methods. We ended up going with gene therapy because it was potentially simpler and potentially more effective. That is, a one-time gene therapy could provide lifelong protection. And there's no need to remember to take a medication every day because the gene is there expressing it continuously. So we went with gene therapy in this first human clinical trial of gene therapy in the brain for any indication. And this was called, so again, this was nerve growth factor or NGF in Alzheimer's disease. These cell types we've been talking about that are deeper in the brain that project the memory circuits of the brain to regulate their function, they're called these cholinergic neurons. And we know that nerve growth factor potently prevents the death of these systems and stimulates their function in animals. So the idea moving forward into this clinical trial was that NGF could do the same thing in Alzheimer's disease, thereby slow the rate of cognitive decline, and who knows, maybe improve cognitive function. Now, right off the bat, though, there was an issue with this approach, and that was that there are other systems in the brain that degenerate too. There are all the cortical cells that are degenerating, and nerve growth factor doesn't prevent their death. It prevents the death of these cholinergic cells that regulate the activity of the cells up here. But would the stimulation of the activation of these cells alone be enough to improve cognitive function in Alzheimer's disease to a meaningful and detectable extent? So that's what we tested in this clinical program. A lot of investigators at UCSD were involved that are listed here. This included neurosurgeons here and at Rush University in Chicago, and eventually at other institutions. This became a program all around the country. And it was supported, our larger last clinical trial was supported by the National Institutes of Health. And again, the rationale was that we were going to target these cells deeper in the brain that send their connections up to the brain, and that doing this would improve cognitive function. I'm going to skip through these slides and just go to the results now. So one way one tries to measure whether you're having a benefit in a clinical trial is by doing a PET scan of the brain. PET is short for positron emission tomography. It involves injecting a radioactive substance into the blood that goes to the brain and lights up. It shows you brain activity if you use a radioactive, if you use radioactive glucose. So glucose is the energy source for cells in the brain. And if you inject radioactive glucose, then you can see it on the radioactive detector of the brain. And this is what a PET scan looks like, doing this kind of glucose PET scan. So blue is an area of less activity, red is an area of more activity, and yellow and green are sort of in between. So these were what the PET scans looked like in our first clinical trial of patients before they received, these are Alzheimer's patients, early Alzheimer's patients. And by early, I mean early in their disease, not young. So these are patients who just received a diagnosis of Alzheimer's disease. And we're going to early patients because that's where this therapy stands the best chance of having an effect before the cells have undergone complete death and loss. So we injected NGF and we did PET scans. And this was the baseline PET scan before undergoing the gene therapy. And this was the average PET scan 6 to 8 months later. And as you can see, there's more red. So it looks like there's more activity overall. We then looked at cognitive function. And this was in our very first trial in just 8 patients. And these are data from the first only 6 patients who underwent bilateral gene therapy. And this is a scale that measures memory and overall cognitive or intellectual function. And low on the scale is worse, and high on the scale is better. And this is the amount of worsening in the 1 year before receiving the gene therapy, in the next year after gene therapy, and the next 6 months after that, then in year 2. And what you can see is that overall, patients were worsening more slowly. However, there was no placebo control group, and this was not a blinded trial. Everyone knew that the patient had undergone gene therapy. So results like this aren't reliable. The most we learned from this was really that, well, we didn't seem to be doing any harm. So we can probably go on to a bigger clinical trial that includes a placebo group. So that's what we did with Paul Azen. We did a phase 2 trial, as it's called, of AAV2 NGF in Alzheimer's disease. This was a multi-center placebo-controlled trial. So in this trial, half of the patients went to the operating room, had a hole placed in the skull into which the vector could be injected, but wasn't. And the other half underwent the same procedure and did receive the vector. In the placebo patients, the needle did not penetrate the brain. But if the patient woke up and subsequently felt their scalp, they all had holes, so they couldn't tell whether they got the therapy or not. And so that's how you do a placebo control in a surgical trial. And this was done at many sites around the country in a total of 49 patients, and half got NGF and half were placebo. I see that there's some questions. Nellie, you want to stop here and take them, or?
Yes, yes, Professor. We
Okay.
had one about the eyes that we can tackle at the end. Right now, I will go from the last one. This was the question, has this gene therapy been considered for Huntington's disease?
Okay, why don't we come back to that one at the end?
Okay,
It's a great question. And yes, the answer is yes, but I can give more detail later.
thank you. And then there is another one. What's the difference between gene therapy and gene editing?
Yeah, that's a great question. So gene editing generally means that you take a gene that is abnormal and you remove it and replace it with a normal copy of the gene. And that's done with a different type of technology than this. That's done with something called CRISPR-Cas9, uh, with the CRISPR-Cas9 tool. So that can actually go into cells in the brain or anywhere in the body. And as I say, can remove a gene that's causing problems, or it can be used to insert a new gene, kind of like gene therapy. In a way, it's another form of gene therapy. It just uses a different thing. We use a virus to just insert something new into the nucleus of the cell, but it doesn't enter the actual genes of the cell. Whereas CRISPR-Cas9 enters the genes of the cells and modifies them. But otherwise, it's another means of trying to reach the same objective. I guess I'll leave it at that.
Yes, and the other question, are you, if they are already working with a pharmaceutical, and if you see the treatment coming to the market anytime soon?
Why don't we come to that one at the end?
Yes.
Okay.
And then there was one about the eyes that was, do you have to repeat the treatment or is it a one-off? The treatment about the eyes.
Yeah, we don't know yet. So we're learning more as patients live longer, and it remains unknown whether there would need to be a repetition.
Thank you. And also, why does gene therapy cost so much, and could the price drop as it is adopted by more patients?
Yeah, the answer to that is certainly yes. The price certainly could drop as it's adopted by more patients. It's kind of a side topic, but I'll just say that so the therapy for spinal muscular atrophy, the weakness in infants, was developed at a university in Ohio called Children's National Hospital. It's a children's hospital in Columbus. And the scientists at that institution founded a company. And that company, once they had their success in children, that company was bought by a big pharmaceutical company, Novartis, I believe. And the price that Novartis paid for this company was something like $11 billion. $11 billion. So what the company that pays $11 billion says is that they have to charge a large amount to recoup their investment. And, I mean, on one level, that argument is understandable. On another level, I don't understand why the company had to be sold for $11 billion. I don't know why the company wasn't sold for 1 billion or 100 million. I don't think that much money went into develop. I know that much money didn't go into developing the therapy, but that calculation for the value of the company was made by bankers. So bankers decide what the market will be, what insurance companies are willing to pay. And on that basis, the bankers say, this is what we think your company is worth. Don't sell it for less than that. And so the big pharmaceutical company comes along. They're told by the bankers, well, it's going to cost you $11 billion to buy this. And what are you going to do? When there are other companies that in competition with you to buy it, they pay it. And then they charge these astronomical amounts to recoup their costs. But at some stage, someone could say to the bankers, well, folks, yeah, if somebody charged that much, they could make the company worth that much, but maybe it's not right to charge that much. Maybe it should cost less. And maybe $100 million is a fair price for this. Or maybe 500 million is a fair price for this. But how do we get to $11 billion? So that's how those prices got to be that high. And you know, if you're talking though about a disease like Alzheimer's, where it affects so many people, then clearly it would bankrupt the company to pay that much for every patient, and it just will never happen. So the cost of such a therapy must be far less than that 2.1 million that's charged for the weakness in children. Okay.
Thank you, Professor. And one more question. When you mentioned that some of the protocols were approved in Europe and not here yet, are we usually a little behind, or how does it work and why is it different, please?
Yeah, well, so there's a European drug agency that's like our Food and Drug Administration. And they, you know, they listen to our FDA, and, you know, often things are submitted for approval in this country before Europe. Sometimes they're submitted at the same time, and sometimes they're submitted first in Europe. Tends to depend on whether the person doing the clinical trial is European or American. And, you know, in the case, sometimes the two agencies just disagree. It doesn't happen often, but it does happen occasionally. And one of the indications in Europe, it's an inborn error of cholesterol metabolism. The European agency thought it was compelling data, and it was the very first gene therapy product ever to be approved. And in our country, the FDA looked at it and said, it's not that convincing. We're not that convinced by the data, and we're not going to improve it. That usually doesn't happen. You know, usually the two agencies are in sync.
Thank you. We have one more question, but we can keep going and make another pause if that's okay with you.
Sure. Okay, so you know, so I built up the expectation. So here we went to this phase 2 trial in a total of 49 patients in Alzheimer's disease. Some got the nerve growth factor, some didn't. So it's really the proper way to do a study. And I showed you the PET scans. And now I'm going to show you some of the tissue results, because at the end of the study, we got brains from many of these patients. And so the brains taught us something. So when the injection site in the brain, which I'm showing you here, was in the right place and expressed the nerve growth factor, so this dark stuff is nerve growth factor protein around the cells that we're trying to target. So when we did that, we found that the cells became larger. Just about 5% larger, but they're still larger. This is called a trophic response. It's a growth response to the presence of the growth factor. And this was important because up to this point, we had no idea if growth factors in the Alzheimer's brain would do anything, if the cells would have the biological tools to actually respond to the growth factor. And this told us that they did. Moreover, we learned that after 9 years in the human brain, these viral vectors continue to make the growth factor. So the gene product is expressed. So this black smudge down here is in, again, the part of the brain that we were targeting. And this is the nerve growth factor protein. So even after 9 years, it continues to be made. So that's good. And we also saw, I'm going to go back to this slide, that when we delivered the growth factor here, that the connections between cells sprouted, they grew toward the source of the growth factor, again, indicating that we're getting the biological response to the growth factor that the animal studies had predicted could happen. So that's good news that, okay, biologically, the Alzheimer's brain can respond to the growth factor in the expected way. So did it improve cognitive outcomes like the smaller first study without placebo controls that I showed you? And the answer is no. So there was no detectable benefit comparing the treatment group to the placebo group in the outcomes. Neither of these changes is statistically significant. This was reported in JAMA Neurology in 2018. So this clinical trial failed to meet its clinical endpoint. So now what do you do? Well, what we did learn is that the cells in the brain, if targeted properly, exhibited these classic so-called trophic responses and cell activation through the expected biochemical pathways. So you know that Alzheimer's brain can respond, that you can have expression for at least 9 years, but there's no benefit in the small phase 2 trial. So why do you have no benefit? That could either be because we had the wrong target. It could be that these cells alone are not a sufficient target as the other parts of the brain are degenerating. It could be that, okay, this mechanism just isn't going to work. But it could also be that you failed to engage the target in enough places to actually give this a chance to show a benefit. And indeed, pathological analysis of about 10 brains showed that most of these injections missed. So this is an example of a miss. These are the cells in the brain that we were trying to reach with the nerve growth factor. Here's another example, and the needle didn't go far enough. And this was the case, in fact, in about 40% of the sites. The targeting was off. And about 60% of the sites were pretty close to the target, as shown in this brain. But among those who were accurately targeted, there was another problem, and that's that the growth factor didn't spread as much as we expected it to. So we expected that the spread from this injection site I'm showing here would be about 3 millimeters, which would bring it down to here. And it would bathe most of these cells in the growth factor. But in fact, as you can see here at higher magnification, the spread was only an average of less than 1 millimeter from the injection site. There just wasn't enough spread. So when you combine that 40% of patients were off target and 60% didn't have enough spread, it nearly failed in most of the sites. You know, that 100% doesn't add up to 100%. That 60 plus 40 doesn't add up to 100% failure because there was some of the injection sites that were mistargeted also didn't have much spread. So overall, about 80 to 90% of the sites just didn't reach their target. So when you reached the target, you had these cellular effects, but when you didn't reach it, you didn't have those effects. So there wasn't adequate targeting. So what did we learn from that clinical trial? Did we learn that NGF doesn't work? No, we learned that we had to target better. That doesn't mean it will work if you target better, but we just didn't end up asking the proper question at the end of the day. So we needed better targeting. So how do we improve? So to move forward, we have to improve the targeting. And that's what we had been working on since that clinical trial ended. And we worked on technology that was really developed by our collaborator at UCSF, Chris Bankevich, who moved to Ohio. And so Chris developed real-time targeting of brain structures for gene therapy with an MRI-compatible needle. So what I mean is that the patient goes into the MRI scanner, we have an MRI-compatible needle, and that can confirm that you have actually placed the needle in the intended site, number one. Number two, we also had to get the growth factor to spread better. So there is a technique called convection enhanced delivery. If you put the flow rate of the fluid entering the brain at a high enough level, then it will show extra spread through the tissue spaces of the brain. And that's called convection enhanced delivery. So Chris developed these methods of convection enhanced delivery in his applications, and we adapted it with him for our target in the brain, the memory circuitry of the brain. The third thing that Chris did was co-infuse an MRI dense marker called gadateridol. This is what's called a contrast agent. When it pumps into the brain, you see it show up on the MRI scan. So not only can you see where the needle is, but now you can see where the gene therapy vector is, and you can see how it's spreading in real time. And if you're off, you can make a correction. If you don't have enough spread, you can continue pumping until you get enough spread. And this can revolutionize the accuracy of these gene targeting methods. So that's what we're doing now as we move forward into another clinical trial in Alzheimer's disease with a growth factor. But we have also switched to a new and potentially more promising growth factor called BDNF that will deliver into a different part of the brain, the specific memory circuitry of the brain. So let me explain that to you. And these are some of the folks that are participating in the current clinical trial. I'd like to acknowledge how important all of them have been in moving this forward. So this was approved by the FDA to move forward into humans in September of last year, and we started enrolling patients in this clinical trial in April. And let me explain what the basis of this is. So BDNF is another growth factor called brain-derived neurotrophic factor. This, in fact, was the second growth factor discovered in the nervous system about 35 years ago. So the entorhinal cortex is part of the brain that's involved in forming memory. It's the area where Alzheimer's disease begins. The entorhinal cortex is the region of earliest cell loss in Alzheimer's disease. It's the site where this amyloid plaque formation occurs. This entorhinal cortex is the memory circuitry for forming new memories. It's the so-called substrate of memory formation, and is a key system with a part of the brain called the hippocampus for forming memory. So this is essential for memory, and memory loss is the cardinal symptom of Alzheimer's disease. This is the region of the brain where Alzheimer's disease pathology begins, and it spreads from there. The BDNF levels decline in this brain region in Alzheimer's disease, and this brain region responds to the receptor, expresses the receptor to BDNF called TrkB, which is necessary for BDNF to show its effects, its biological effects. So putting this all together, we say, let's try to give BDNF to this area of the brain where Alzheimer's disease first starts in an effort to see whether we can slow memory loss or possibly even improve memory. So how does this differ from the first trial we did with nerve growth factors? So first of all, nerve growth factor, it was a different growth factor, and it targeted these cells deep in the brain that send their connections to the cortex where cells are also degenerating. But BDNF is the growth factor of cells in the cortex. The problem with NGF is that NGF didn't directly prevent the death of these cells. It only aimed to excite them better. But BDNF is the growth factor of these cells that can prevent their death, as we showed in animal studies, and I'm about to show you. So in contrast to NGF, BDNF is a direct growth factor for neurons degenerating in the cortex. And in animal models, it prevents the death of these cortical cells, it stimulates their function, and it builds new connections between the cells. So let me show you what some of these animal studies look like. So one of the most common animal models of Alzheimer's disease is the so-called Alzheimer's mouse. So this is a mouse that expresses two gene mutations that are associated with Alzheimer's disease in humans. So there are some gene mutations that cause Alzheimer's disease and early Alzheimer's disease in people in their 30s and 40s. It's not the garden variety of Alzheimer's disease that occurs later in life. These patients have mutations in the amyloid gene, and they develop early Alzheimer's disease. So using techniques of genetic engineering, you can engineer mice to express these human gene mutations, and they form amyloid plaques in the brain. So we took these mice and we gave them BDNF gene therapy starting at age 7 months after the onset of the disease in the mice. These mice lived to a maximum of 24 months, probably less in nature, but in the laboratory, they lived for 24 months. So we started the therapy after their disease onset. We let them express it for 3 months, and then we tested the learning and memory, and then we took the brains and examined the effects on cells. So this is the learning and memory results of this study. So this is putting these so-called Alzheimer's mice, their scientific term is APP transgenic mice, into a learning and memory test. So the way this is done is that the mice are placed into a pool of water, and they can escape the water by climbing onto a platform that's located in one part of the pool of water. And the mice know how to swim, but they don't want to be swimming all the time. They want to be standing on something and escape the water. So they can learn the location of this escape platform in the pool by looking at cues in the environment. They have to remember where the spatial cues are to be able to escape the platform. When you take a normal mouse, shown in green here, then on the first day of testing, this is the distance it swims before it finds the platform. And when you expose it over subsequent days, it locates the platform after swimming a shorter distance because it has learned, it has formed a new memory for where the platform is. Similarly, you can take the time that it takes to locate the platform, On the first day, it takes about 35 seconds of swimming. And by the last day, it's down to under 20 seconds. So this is normal learning. If you take these amyloid mice, they're worse to begin with. And it's not because they can't swim and they have weakness. We check for all of that. They don't. They're just not as good at locating the location of the platform, but they swim equally fast. And they also learn, but their learning is never as good as the wild type mice. And so they have impaired memory, which you might expect from an amyloid model. But now if you take these same mice and you give them the BDNF gene therapy, they're similarly impaired at the outset, but they learn better. So, and they also find the escape platform faster than the mice that didn't have the treatment. So this improved their memory. And there's another memory test called contextual fear conditioning. I won't get into it other than to say that the amyloid mice, once again, are impaired compared to the normal mice, and BDNF treatment corrects the impairment in these amyloid mice. There's another version of this test that doesn't depend on memory, and there's no difference between groups, as you would expect, because we didn't treat this other part of the brain. Now, if you look at the connections between cells, that's called the synapses, and we can look at that with a label for these connections, that's called synaptophysin. Then the Alzheimer's mice have a reduced intensity and number of these green connections compared to the Alzheimer's mice that received BDNF. And if you compare these groups again, the normal mice, the Alzheimer's mice, and the Alzheimer's mice treated with BDNF, you see this correction in these synapses, the connections between cells in the entorhinal cortex, where we do the gene therapy, and at its downstream targets in the hippocampus also. So this beneficial effect is present both where we deliver the gene in the entorhinal cortex, but the benefit is transmitted into its partner structure, the hippocampus. And that's because the BDNF protein is transported down the connections from the entorhinal cortex to the hippocampus. And then this is looking at how genes change as a function of this amyloid mutation. So everything red here is a gene that has been turned on by the presence of the amyloid mutation compared to a normal mouse. And everything green are genes that have lost their expression as a function of having the amyloid mutation. And now many of these changes are corrected here. So black means you're kind of normal. You're at the level of an intact mouse. So things that went up here now are being corrected by the BDNF treatment. So things that were up are no longer up. Things that are down have been corrected by the BDNF gene. Overall, about 2/3 of the genes that were perturbed as a result of the amyloid mutation have been corrected by the BDNF therapy. And that's true both in the entorhinal cortex and the hippocampus. And this just shows that the BDNF protein is transported from the entorhinal cortex into the hippocampus. I don't think I'll talk more about that in the interest of time. Now, these mice also show a loss of cells, a death of some cells over time. And if we treat them after disease onset, we don't see a change in those cell numbers. But if we treat them before disease onset, we do see some protection. So this is the death of cells in the amyloid mice compared to normal mice at the time that we take the brains. And if you give them the BDNF, then you see an improvement in that survival of cells if you give it early enough. Hence the rationale for going early in the disease. So that was one set of findings in these so-called Alzheimer's mice. We also looked at aged rats. So rats with aging, some types of rats show cognitive impairment with aging. So we had a total of about 70 rats in this study. We took young animals and we took aged animals that either had cognitive impairment here or didn't. And those animals with cognitive impairment, we gave BDNF. And we assessed the effects of that on their learning and memory, and then we looked at the cells. So this is data like I showed you for the Alzheimer's mice. These are young rats in the same pool of water. And as you can see, if you look at the distance they swim, this is how much they swim on the first day, but they learn where the location is, and so they don't swim as far. This is the time they take. Again, the time gets better over time. Now, if you look at the aged rats, they all start out about the same as the young rats, but they just never learn as well. So this is before we give them BDNF. Now, if you test them again after giving them BDNF, here's the performance of these BDNF-treated animals. And it's not normal, but has improved significantly compared to the rats that didn't get the BDNF. And we see this improvement both on the distance and on the number of seconds it takes them to locate the hidden platform. So again, just with age-related memory impairment in rats, we see a benefit with BDNF. I'd like to emphasize, if you give the same kind of a task to humans, we humans don't show this kind of decline with aging. We show far more mild cognitive decline. These are rats that have been inbred for generations and generations and generations. So, we humans don't tend to inbreed that much anyway. And if we did, we would probably have similar impairment with aging, but we don't. So we're not as bad off with aging. These rats are especially bad. And if you give them BDNF, they improve. Once again, if we look at all the genes that are perturbed as a function now of aging in these inbred rats, then we see that with aging, some genes are turned up. That might be damaged genes. Some genes are turned down. These might be good functional genes. And again, if you give these aged rats BDNF now, there's a normalization of many of these changes. And it improves, in this case, about 42% of those changes. So I'm not going to go through all the results. I'm going to skip ahead now to the monkey results. So based on those findings in lower species of animals, we said, okay, this is looking interesting. This could be a candidate human therapy. Let's bring this as close as we can to humans before we try this in humans. So these are rhesus monkeys. And we took the same part of the brain, the entorhinal cortex, and we cut the connection from the entorhinal cortex to the hippocampus. That's a lesion model. So when we cut that connection, these cells in the entorhinal cortex degenerate. So these are the normal cells in the entorhinal cortex, nice big plump cells. And when we cut their connection to the hippocampus to impair memory, then you see a degeneration of some of these cells. You have a sense that there are fewer big cells here. And when we treat with BDNF, you have a sense something is going on here. It's not normal like the intact animals, but you think there are bigger cells here. So when you rigorously quantify this effect, here's what you see. This is the normal number of cells in this region in an intact monkey. If you do the lesion, you lose about half the cells. And if you give BDNF, you protect about 80% of the cells. So that's, again, a sizable effect. And if we look in the monkeys to see whether the BDNF is being transported after you inject it in the entorhinal cortex into this hippocampus, the other half of that memory circuitry, indeed it is. There's more of this dark staining here in monkeys that had the BDNF vector injection than in uninjected animals. So the BDNF is being transported throughout the memory circuitry. So then we took aged monkeys. So aged monkeys have some cognitive impairment with aging. It's not as much as humans, but there's some cognitive impairment. And so we tested it in aged monkeys. So this is the entorhinal cortex in an aged monkey. And this is an entorhinal cortex in an aged monkey that got BDNF. And again, you have a sense that there's a difference here, that there is more intense staining and maybe more cells. And there aren't more cells, as it turns out, but if you quantify the size of the cells, there's about a 20% increase in cell size. Again, this is a classic trophic effect. So does this kind of thing translate into a benefit on memory? How do you test memory in a monkey? So it's kind of neat, actually. So you show the monkey a computer screen, and it sees this one purple dot. Then the screen goes blank, and it sees another screen with this yellow triangle. Then there's a delay of seconds to minutes. Then the monkey sees this screen, and it has to touch the screen on the location of the original dot. And then the screen goes blank, and then it sees this screen, and it has to touch the screen on the location of the original dot again. And if it does it, then it gets a sweet food reward. It's motivated by the food reward. So before undergoing gene therapy with BDNF, before gene therapy, this is the performance of young monkeys. This is the performance of aged monkeys that will not go on to get BDNF. And this is the performance of aged monkeys that will go on to get BDNF but haven't gotten it yet. So you see the two groups before BDNF are equally impaired. But now if you give them BDNF and check their performance again, there's this improvement in the monkeys that received BDNF. So there's cognitive improvement in aged monkeys. So this was the rationale for moving this to human trials. In amyloid mice, in cell culture, in injured rats, in aged rats, in injured monkeys, and aged monkeys, this all improved cellular measures of function and in some cases cognitive performance if the problem caused a problem with cognitive performance. BDNF was transported into the hippocampus, and we saw consistent effects on the activation of cells, on gene expression, on synapses, cell survival, and learning and memory. So this all supported moving this forward, but we had to move forward this time with these improved targeting methods. So now we're using this real-time MRI targeting. We're doing this convection enhanced delivery and this GATA-pteridol co-infusion so we can see exactly where the needle is and if it has delivered the expected dose of the growth factor. I see we're at 1:30, so I guess I ought to be wrapping this up. So let me wrap it up. Here's the MRI compatible needle going into the right part of the brain. Here's the infusion of the growth factor over time. And here, when we take the monkey at the end of the study, we see that we have delivered the BDNF where we intended to. This is where the MRI showed it would be, and that's where it is. So we're moving this forward into humans. There will be two sites. One is here at UCSD, the other is at Case Western. We'll do the gene delivery by Dr. Bankevich and his colleagues at Ohio State for this first study because they have the most experience with this. It'll be done in 12 patients, 6 with early Alzheimer's disease, and if it's safe, we'll move on to 6 patients with mild cognitive impairment. And we'll focus on clinical endpoints that assess memory. And we'll test a couple of dose groups. And the whole rationale for the study is that if we go in early patients, we'll see better neuroprotection. So I think that's a good place to wrap up. I'll just wrap it up there. So our objectives now are to complete this phase 1 study. And if this shows safety and a potentially positive signal, then we'll move forward into larger phase 2, 3 multi-center clinical trials with placebo controls. But the whole basis of this program is that we need to do better in Alzheimer's disease. The existing experimental therapies just have not been booming successes. And of these potential therapies, growth factors are the only ones that have the potential. It doesn't mean they'll do this, but they have the potential to improve cognition. They also have the potential, as the other therapies do, to slow the rate of cognitive decline. But the hope is that we can, perhaps if we're fortunate, even improve memory with this approach. So I thank you for your long attention. And if there's any time left, I'll be happy to answer more questions.
Thanks to you, Dr. Tuszynski. Thank you very, very much. I have questions here in the chat. In the context of AD, how is aging defined? I have heard that people even under 40s develop.
I'm sorry, can you say that again?
In the context of AD, how is aging defined? I have heard that people even under 40s develop.
Yeah. So a young person can get Alzheimer's disease if they have a gene mutation causing it. So that's what we're looking at in those mouse studies. So there are several known human mutations that result in early Alzheimer's disease where it can have its onset in the 30s and 40s. And a lot of those families actually live in Colombia. So there are clinical trials being done in Colombia in those families to see if treatment before they develop symptoms of Alzheimer's disease will be useful. Because the idea has been, since so many of the other therapies have failed in patients already with the diagnosis, maybe they really need to be treated very early in life. And since so many of those patients are in Colombia, a lot of those clinical trials targeting amyloid are being done in Colombia. But the most common form of Alzheimer's disease occurs with aging. And as the older we get, the higher the proportion of people there are that end up developing Alzheimer's disease. And that's a different disease in the sense that it's not caused by a single gene mutation. I talked about the ApoE4, and that's a gene, but it's not a mutation. I know folks on this call have a lot of different backgrounds. Some know genetics and some don't, but we all have we have two copies of every gene, one that we got from our mother and one that we got from our father. And those two copies can sometimes come in slightly differing forms called alleles, A-L-L-E-L-E, alleles. And those two copies of the gene, those alleles might be slightly different depending on the gene. And so one of our 20,000 genes is called the ApoE gene. That's short for apolipoprotein E. And it comes in actually 3 types. And the types are called, well, they're not called 1, 2, and 3. They're called 2, 3, and 4 for reasons that escape me. But anyway, you can get either the 2, the 3, or the 4 copy of this normal gene. But it turns out that if you get 1 copy of type 4, you have a higher risk of getting Alzheimer's disease. It's not a mutant gene, it's just an allele. It's a different form of it. But having 1 of those genes gives you a higher risk of developing Alzheimer's disease. If you have 2 copies of the E4 allele, you have a much higher risk of developing Alzheimer's disease. If you don't have either of the E4 alleles, you have a lower chance of getting it. By the way, when you do 23andMe, they tell you whether you have any of these copies of the E4 allele. So for $199, well, you can find out, and a lot of other things too. I do not have any stock in 23andMe, so that's not an endorsement. But anyway, so with aging, we all have a higher risk of developing Alzheimer's disease. If you add your type of ApoE gene on top of that, that further defines your risk with aging. So I hope that answered the question.
Thank you. And a reminder, please, about the Huntington's disease question that we were asking.
Yeah, so the question was, are there gene therapies being developed for Huntington's? And the answer is yes. And there are, I believe there's a clinical trial underway now delivering a growth factor gene to Huntington's patients to see if that will be neuroprotective. So that growth factor is called GDNF. It was one of the growth factors also tested in Parkinson's, also being tested in Parkinson's disease. So yeah, that's one approach. There's another genetic approach. It's not a gene therapy per se. It's an antisense oligonucleotide. So that's another, breakthrough therapy in molecular biology that has now been shown to benefit the human condition. And there's this company in San Diego called Ionis. And again, I'm collaborating with Ionis on a project in spinal cord injury, but I have no financial interest in Ionis. But they were the company that developed a technology for what are called antisense oligonucleotides. So these are fragments of a organic compound that block messenger RNA in cells. So if folks remember, we have our genes, that's the DNA. The genes get so-called transcribed into RNA. RNA, this all happens in the cell nucleus. RNA goes from the nucleus into the cytoplasm of the cell, where the RNA is made into protein. So you go from DNA to RNA to protein. And antisense oligonucleotides interfere with making the RNAs, that intermediate step. And they have chemical compounds that can block those messenger RNAs and take out deleterious things. And one of those deleterious things is the Huntington disease gene. It's the so-called Huntington gene. So there's an antisense oligonucleotide trial ongoing for Huntington's disease. And it's an interesting trial. I'm not aware of any results yet, but it's taking advantage of that technology. And it's really pretty impressive technology. And it has already shown a benefit in what disease? Spinal muscular atrophy, the one in which there's a gene therapy benefit too. And I don't know if what I'm about to say is correct or not. And I know this will be publicly posted, so nobody, fact checked. But I believe that the gene therapy is more effective than the antisense oligonucleotide therapy. But the gene therapy is, for a fact, much more expensive. So anyway, that's another therapy that has come from the era of molecular biology that could really transform how we treat human disease. And if I may, let me just say another word about that. You know, you folks are probably aware that there are a lot of antibodies now that are useful therapies for disease, Herceptin, and other monoclonal antibodies for a variety of cancers, for rheumatoid arthritis, for multiple sclerosis. We just use a lot of antibodies these days. You know, I was a medical intern, when that technology first came to medicine, and they were all failures at first. For the first 10 and 20 years, all these monoclonal antibodies, everyone said, wow, this is going to transform medicine. And they were abject failures for the first 20 years. And then the first approval came in cancer, and then the next, breast cancer, and then the next, and the next, and the next. And now they're incredibly valuable tools for treating medicine. And I think that's where we are now. We're at that same precipice where just the first approvals have come in gene therapy, just the first approvals in antisense oligonucleotides. And I do think that this will be an opening into far more successes in treating a variety of diseases. And that's why these pharmaceutical companies are paying tens of billions of dollars to have access to this technology. And something more sensible has to happen there, because these prices are untenable and unethical.
Thank you, Professor. One more question. Do you have access to UCSD bodies donated for studies in the body donation program?
I'm not quite sure how to answer that. What I can say is that when patients have enrolled in our clinical trials of gene therapy here, most of them have been kind enough to donate their brains to us. And the last time I got one of those brains was yesterday. So, you know, having access to that material is so incredibly valuable. And you saw one example in what I said today. We did gene therapy in those first sets of patients with Alzheimer's disease, and we didn't see a clinical benefit. If we hadn't gotten those brains, we wouldn't have known that our injections missed. And if we hadn't gotten those brains, we wouldn't have known that actually at the cell level, when we hit the target, this works. So that knowledge combined allowed this program to find its next direction, which was to use better targeting. And that's how incredibly important body donation is. You know, the ability, you know, the gift from those patients, first to participate in the trial, and then secondly, to donate their brains, was just fundamental to this moving forward. And we hope, you know, in the future, identifying a useful therapy.
One more question, if this is okay with you, Professor. Why in countries like India, Alzheimer's disease is rare?
I don't know. So I don't know if I don't know if that's factually accurate, and perhaps the person posing the question does know. I just don't know. But the first thought that comes into my mind, not knowing, is, do folks live as long in India? What's the average life expectancy? Because this is clearly an age-related disease. So if people don't live as long, the same percentage of the population will not have it. Another thing I can say is that, you know, India has certainly made incredible strides in the last few decades in becoming really a predominant middle-class country, and no longer a poor and an uneducated country. And there has always been the intelligentsia in India, and many impressive things historically have come from India. But there was still the fact that much of the population couldn't afford to go to school and lived in poverty. Not all, but, you know, a proportion. And a study done at UCSD now about 25 years ago by somebody named Robert Katzman, who went to China at that time, just as China was opening up, and looked at Alzheimer's disease in China, allowed them to study a huge number of patients, and found that the amount of education one has reflects the risk of developing Alzheimer's disease. More education is protective. And the reason appears to be because more knowledge, more study, more thought builds more synapses in the brain. And that provides a reserve that we can fall back on when things start to degenerate. If you have more connections, you have a reserve. It's like extra fuel in the tank. And so, you know, when you ask the India question, I think, well, How much education is there generally in the society? How long do people live? Are there other things that could affect the risk, like diet? Aluminum in pots. If you have ginkgo in your diet, is that protective? These are all questions that have been asked and studied, and most of the answers and the best studies we have seem to indicate there's not an effect of aluminum in pots, or that ginkgo is not helpful. But who knows? There's much more that we don't know than there is that we know for sure.
Right. That was precisely the next question, that if there are studies that aid in suggesting what people could do to prevent, but you just answered that.
Well, but there is another answer, actually,
Okay.
that I actually think is helpful. And this is the reason I exercise. And that's the potential benefit of exercise. So studies have shown a couple of things. If you have an active support group of people, if you remain active as you become elderly, that appears to be protective. And there's better evidence to suggest that than anything else we've talked about up to this point. So first of all, if you subject just mice to physical exercise, a number of studies now have shown that that increases levels of what? BDNF in the brain and in the hippocampus and in the circuitry that we're targeting. So exercise increases BDNF. An enriched environment in mice increases BDNF. And so there's this thought out there, unrelated to any of the work that we've done, that BDNF is protective and that exercise makes it more available. And there are some epidemiological studies that support, but they're not that clear. Epidemiological studies in tens of thousands of people are just tough because they depend on reporting and reliability and follow-up. So it's always tough. But epidemiological studies are kind of equivocal on whether more activity helps. But there have been some studies that suggest that an active social network helps, that continued engagement, just as everyone is doing with this call, that helps, and that physical activity helps. And when I talk to patients about this and they ask, what can I do? My answer is stay as active as you can, stay as socially engaged as you can. It certainly can't hurt, and there's a good chance it does something.
Great, thank you. And one last question, please. Are the researchers being paid by grants or contracts by the federal government?
Well, often, yes, that's the usual answer. So I'm faculty here at UCSD. Part of my salary comes from the state of California, supporting positions for faculty. So that's part of my salary. There are a number of faculty here who don't have that line of support from the state. And then in that case, their salaries have to come, typically from grants. One can also have contract from companies. We don't have those. We do not have those. They can come from private foundations. So some of our funding comes from the Alzheimer's Association. Some of our funding comes from philanthropy too. That's played a role. For example, folks here in San Diego probably know the Shiley Family Foundation, Darlene Shiley and her late husband, who have been generous benefactors in this community. And they made a donation to support this clinical trial that we're doing now. And she also supported, she and her husband at the time, he died. He was still her husband. But the Shiley family also supported our very first gene therapy trial in humans, that first nerve growth factor trial. So philanthropy also plays a role, and it shouldn't be understated. Most of our funding comes from the NIH to support this work. And our current clinical trial is NIH, the Alzheimer's Association, and the Shiley family.
Great. Professor Tuszynski, thank you so much for your time and your very valuable information. We really appreciate you being here. If we were live, you would be hearing the clapping. So thank you so much. We really appreciate it.
It was great to have the chance to talk to everybody. Thanks for your attention.
See you next time. See you in October. Thank you. Bye-bye.